All right, welcome everyone. I hope you had a great weekend. Got some sun, not too much. Got some warmth, but not too much. No one got heat stroke or too much sunburn. So we're going to continue today to talk about metabolism. And I just want to remind us of where we were when we left off. We were comparing aerobic respiration, which is a metabolic process that is performed by lots of organisms and definitely should have been drilled into you already because it is the one we humans use. And it's comparing where the different parts of aerobic respiration happen in a eukaryotic cell, which you would have studied before, and a prokaryotic cell, and also comparing the ATP yield, noting that in a prokaryote, there's more ATP because of the lack of a membrane. So there's no need to spend ATP bringing chemicals into the mitochondrion. And I showed you this little diagram. I'm just going to click it through really fast, really kind of, again, comparing prokaryotic respiration to eukaryotic respiration, all the same processes, but where they happen. And the other reason to show you this picture is to encourage you to draw your own and not to worry about not being a good artist. As you can see, this is really a terrible drawing, but it helps me remember the important components of this. And I suspect for many of you, you'll find the same. The next comparison we need to make so that we can better understand all of the diverse metabolic options in microbes is we need to compare aerobic and anaerobic respiration. So sometimes people have studied this in the past, often they haven't, and there's also sometimes misinformation that's taught accidentally. So we really need to make sure that we're clear on what the differences are and what it isn't. So these two processes are similar in a number of ways, sort of biochemically in terms of what is required to make them happen. They share some metabolic components. Okay, so if we think about aerobic respiration, we started with glycolysis, you go through the Krebs cycle, you go through the electron transport chain. Now, anaerobic respiration actually uses that exact same series of steps. That part is not different. The difference comes in what regard? How would you put into the chat, how would you summarize the difference between aerobic respiration, which goes through glycolysis, the Krebs cycle, electron transport chain, and electrons end up on oxygen, and anaerobic respiration? What would you say is different? Great. So I see two answers in the chat, both of which are correct, and they're surfacing exactly the important things to know about the differences between these pathways. So while they share these metabolic steps, the very final step, where the electrons go after they exit the electron transport chain, which of course in aerobic respiration is they go to oxygen. In anaerobic respiration, by definition, there is no reliance on oxygen. It's a process that can run without oxygen. It's not to say that it can't run in the presence of oxygen. It's not that oxygen is inhibitory, but rather that it doesn't need oxygen. And instead, those electrons go somewhere else. So one way of saying that, and it's jargony, okay, but I'm gonna explicitly call out the jargon because it's important for you to know what this means, is the terminal electron acceptor is different. The terminal electron acceptor is where do the electrons go at the end of the electron transport chain. And the terminal electron acceptor is not oxygen. In anaerobic respiration, it's something else that's similar to oxygen in its biochemical properties, in its ability to pull on and accept very low energy electrons. So something like nitrate, but also carbonate and sulfate would be examples of alternatives that could be used in anaerobic respiration. And as some of you mentioned, and this is also very important, actually anaerobic respiration is not as good at making ATP. It's better than fermentation, as we'll see in a moment, but it's not as good as aerobic respiration. So of the possible ways to get ATP from oxidizing glucose, aerobic respiration is superior. So we're going to see what's going on here. Again, another little diagram. So what I've drawn now is prokaryotic because this doesn't really happen in eukaryotes. So I've drawn a prokaryotic cell, and that's why there's no membranes for mitochondria. Everything is happening either in the cytoplasm, the cell membrane, or the extracellular space. And we see that 6-carbon glucose is broken in half to 3-carbon pyruvate. In the process, ATP is made, 2 ATPs. We're not doing any accounting here. And some electrons are removed and put onto NAD+, making it into NADH. It's actually 2. Once pyruvate is made, you do another step to prepare it for the Krebs cycle. You remove a carbon, throw it away as carbon dioxide. That leaves a 2-carbon molecule called acetyl. And you harvest more electrons. And then it enters the Krebs cycle, you make some more ATP, you harvest more electrons, and finally all these NADHs go and drop their electrons off of the electron transport chain, oxidizing themselves back to NAD+. Those electrons travel through the chain, pump protons across the membrane, creating a gradient that can be used to make ATP through ATP synthase. Now, in aerobic respiration, the electrons at the end of the electron transport chain are put onto oxygen, creating water. And in anaerobic respiration, the major difference is that there's just a different enzyme complex to put electrons onto a different molecule, such as nitrate. So in this particular example, this is nitrate reductase, and it's adding electrons to nitrate, producing nitrite. And if we wanted to diagram anaerobic respiration using carbonate or sulfate, we would actually have to modify this diagram again, because remember, these enzymes are very specific. And so this guy can't hand electrons off to nitrate. It can only do it to oxygen. If there's no oxygen, it's not going to do any good. If you want to do this type of anaerobic respiration, you need this specific enzyme. And if you want to use carbonate as a electron sink or as a terminal electron acceptor, you need yet a different member of the electron transport chain. So when we do this with aerobic, when we do this process and it's aerobic respiration, when we have the right enzymes and oxygen is present, we get 38 ATPs, again, in a prokaryotic cell. How much ATP do we get if we're anaerobic? Put that into the chat. If we do anaerobic respiration, for example, it's diagrammed here. Do we get 38? Do we get less than or more than? Put that into the chat. And if you're not sure, put a question mark. It's totally fine. Okay, great. So the answer is less than. We get less than. You get less than no matter what type of anaerobic respiration. And so to begin with, maybe just memorize that. Okay, but let me try and help you to remember it. If you're the kind of person where it's hard to remember stuff unless you understand why it's true, then let me explain why it's true using this diagram. So one thing that you'll notice is where I put this enzyme and what's not happening. So you'll notice that this complex right here has an arrow indicating that protons are pumped, and this one does not. So basically, and I'm oversimplifying the situation here, but this is good enough. Basically, when you modify the electron transport chain and you remove cytochrome C oxidase, whatever alternative you put here, it doesn't pump protons. And so you don't get that last push of protons across the membrane. So you don't get as robust of a concentration gradient, and you don't get as many protons coming back through ATP synthase, and you don't get as much ATP produced. It's still going to be, more than what's made here and here, okay? But it's going to be less than what would have happened if we'd used cytochrome C oxidase or one of the alternative enzymes to cytochrome C oxidase that still does aerobic respiration. Does that make sense? Yes, no, or kind of? Okay. So again, if you are struggling with the why, I'm not going to test you on the why actually, though the why could be very helpful in remembering. But if you do struggle with the why, then just go back to just knowing that aerobic respiration produces more ATP per glucose than anaerobic respiration. That's just a fact that you have to know. Spencer asks a very good question. Spencer said, how much less? The reason I can't give you that answer is that anaerobic respiration is not just one monolithic process. That means, if you're doing nitrate reduction or carbonate reduction or sulfate reduction, actually the modifications to the electron transport chain are all different for each of those terminal electron acceptors. And so it varies. It's not the same. Aerobic respiration is basically the same, right? It's always 38, and that's because it looks like this. There's 3 places that protons are pumped, and that's the end of the story. But for some of these, only 2 times do we get proton pumping, and some we only get once. So it really varies depending on the details of how the modification has been done in that particular organism. Okay, now the next comparison we have to make is between respiration and fermentation. And here, let me see if I can reinforce this. Here I'm saying respiration, and that means all forms of respiration, aerobic and anaerobic. And fermentation, all forms of fermentation, because there's lactic acid fermentation and ethanol fermentation and butanediol fermentation and X, Y, and Z fermentation. So we're comparing all of the respirations to all of the fermentations. Nonetheless, all of these processes, whether we're doing something that's aerobic respiration or anaerobic respiration, or whether we're doing any kind of fermentation, they all start with the same metabolic pathway. And what is it? That's right. They all start with glycolysis. And before I ask this next question, let me ask you a sort of sub question, a sort of memory question. When you do glycolysis, who remembers how much ATP you get from that substrate level phosphorylation? When you do glycolysis, just glycolysis, guys, just glycolysis, not respiration, just glycolysis, how much ATP do you get? Because you do make some ATP in glycolysis. If you were to stop at the end of glycolysis, you would still have a net yield. That's right, you have a net yield of 2 ATPs, okay? So I want you to remember that. So if we compare respiration and fermentation and we say, well, they both start by taking glucose and breaking it in half and getting out 2 ATPs. Now, if we talk about respiration, what happens next is we go through the Krebs cycle, we go through the electron transport chain. In fermentation, do we go through the Krebs cycle? In fermentation, do we go to the electron transport chain? Both, neither, one or the other? That's right. The answer is neither, okay? Fermentation makes no further ATP after glycolysis and does not go through the Krebs cycle. Remember that you make some ATP in the Krebs cycle as well by substrate level phosphorylation, just a little bit. So that doesn't happen. And you make a lot of ATP in the electron transport chain by oxidative phosphorylation. That doesn't happen. So it's not that fermentation stops exactly at the end of glycolysis, but it does stop making ATP. And then to be considered fermentation, what happens next is 1 or 2 additional reactions to sort of tidy things up. Another way of stating the difference between respiration and fermentation is to say that the terminal electron acceptor is different. And we can categorize all of the terminal electron acceptors in respiration under this umbrella. It's some kind of inorganic molecule. Oxygen's an inorganic molecule. This is the chemical term, right? Meaning no carbon-hydrogen bonds. Oxygen, nitrate, carbonate, sulfate, all of those possible electron acceptors at the end of respiration are inorganic chemicals. Where electrons go at the end of fermentation is an organic acid, usually, or an organic chemical of some kind. That means a high energy molecule with carbon-hydrogen bonds in it. And remember that fermentation stops making ATP after glycolysis. So how much ATP do you get after you completed any form of fermentation? Put that into the chat. Even though it's on your screen, I want you to put it in so that your brain can remember this. Right. So fermentation produces 2 ATPs, whether it's lactic acid fermentation, ethanol fermentation, X, Y, or Z fermentation, all of the fermentations, you get 2 ATPs. And again, I am slightly oversimplifying the situation, okay? So that if you go on and become a world expert on, mycology or bacteriology and fermentation processes in the industry, you don't look back and think that I deliberately misled you. But for our purposes, it's fine to say this. All forms of fermentation get us 2 ATPs and that's it. So if we compare that to respiration, any form of respiration is going to be more than 2. I mean, without knowing exactly how much is made in the electron transport chain, at the very least, we know that we got 2 ATPs in glycolysis and 2 in the Krebs cycle also. We at least have 4. And the oxidative phosphorylation events that occur in the electron transport chain are going to give us more than that. So we know that anaerobic respiration is more than 2. And we also know that anaerobic respiration is less than 38. Okay, so I'm going to draw you a little diagram. So I'm showing you here on the left respiration. And I'm abstracting it outside of the cell altogether. So I've just removed all the cell membranes and stuff and just said, hey, this is what happens. We got our glucose, we break it in half, we decarboxylate it, we throw it through the Krebs cycle. Then we take out all these electrons from glucose, from pyruvate, from the various intermediates of the Krebs cycle. We take out all these electrons, we throw them into the electron transport chain, and we punch out a bunch of ATPs, usually 38 if it's aerobic and somewhat less than 38 if it's anaerobic. We also get a little ATP here, a little ATP here. If we look at what's happening with fermentation, this first part is still the same, right? We still go through glycolysis. We still make pyruvate. We still actually take out some electrons. But those electrons on NADH are not going to the electron transport chain, so there's no way for them to be oxidized back to NAD+ and come back and collect more electrons. Okay, so what happens instead is that the electrons are put back onto this product of glycolysis, pyruvate, and that's what oxidizes NADH back to NAD+. Now, there's 2 things to know here. One thing is, you might ask yourself, well, that's dumb. If I'm not going to use the electrons for energy, why would I go to the trouble of harvesting them in the first place? And all I can tell you is, in order to break glucose in half, that's what you have to do, okay? We just don't get to use those electrons for energy. We just waste them, basically. We put them back on the product of what we made. That's the first thing. The second thing to know is that this diagram specifically shows you something called lactic acid fermentation, which our body does, okay? So pyruvate electrons are added to it. That produces a 3-carbon molecule with more electrons than what pyruvate had, which is called lactic acid. This is not the only form of fermentation out there. You may have heard of a little organism called Saccharomyces cerevisiae, also known as budding yeast, and maybe you haven't heard of their name, but you know what they do. They're famous for making bread, beer, and wine. And so those organisms do fermentation, but they don't make lactic acid as a product. They make what? When you're making wine, yeast takes sugar and makes what out of it? That's right. A 2-carbon organic molecule called ethanol. And you may also have noticed that beer and wine is fizzy. The bubbles in the beer and wine are a gas, and that gas is carbon dioxide. How do you achieve that? Well, it just so happens that yeast doesn't actually stop right here and do this reaction. It goes one step further. It does this step here and makes a 2-carbon molecule. And that 2-carbon molecule is where it puts the electrons. And when you add the electrons to this 2-carbon molecule, you make a 2-carbon molecule called ethanol. And in the process, you release CO2, which is why your beer and wine is fizzy. Now, have we described the whole universe of fermentation reactions? Is lactic acid fermentation and ethanol fermentation the only game in town? You are correct. This is two famous examples that have been well studied because we do this and our good friend that makes beer does this. Okay, so that's why we know so much about these. But if you look at microbes that do fermentation, some do this, some do that, some do both, some do neither, some do a bunch of other stuff. Okay, the ways in which these fermentation reactions differ comes down to what exactly the product is. It's sometimes an acid, it's sometimes an alcohol, sometimes it's an acid alcohol, sometimes it's something else, an organic solvent. And the difference between all of these different strategies is just, did you have this enzyme or did you have these two enzymes or did you have something else? Now, does that make sense? Yes, no, kinda. Great. And just to kind of put us in the right mindset for Wednesday, what is the difference? Like, why does one organism use this enzyme and another organism use these two enzymes? Like, how come they have these enzymes? What is it that determines which enzyme an organism is going to have? That's right. Their genes, because enzymes are encoded by the DNA. Okay, so here's a little cartoon from any standard textbook illustrating much better than I have the difference between respiration, all forms of respiration. Here, aerobic respiration is shown, but if you just change this molecule out, you're describing anaerobic respiration, and fermentation. And here you can see that this is being deliberately vague about what happens after pyruvate is made. And so this, from your prep, from the video lectures you already watched, kind of nicely illustrates a small amount of the diversity of fermentation reactions possible in different members of the microbial world. Okay, so some do lactic acid fermentation, and that's their only strategy. Streptococci, strep throat is caused by a streptococcus. Lactobacilli of yogurt fame. Bacilluses, many of you are studying bacilluses right now for your unknown ID project. Saccharomyces cerevisiae, this is the budding yeast that makes beer, wine, and bread. What do they do with it? How does this relate to fermentation? They do ethanol fermentation. Propionibacteria make different things. Clostridium, this is our genus that is an obligate anaerobe, lives in the soil, and includes very severe human pathogens that cause tetanus, C. diff, and botulism. Okay, so here are some of the yummy things that they make when they do fermentation. You can imagine you wouldn't want these in your body. E. coli and its friends, Salmonella of salmonellosis fame, does ethanol fermentation and lactic acid fermentation and a bunch of other acid fermentations and so on. So what we can see is there's lots of fermentations. Different species may do one or the other or a variety of things or none of them. Briefly, I want to walk us through the catabolism of other macros. In terms of microbes, the relevance is that just like you, many microbes can eat more than just sugar. And in order to do so, they need enzymes. And those enzymes we're going to be testing for during our unknown ID process. So again, another diagram of respiration, okay? Glucose broken half to pyruvate, remove one of the carbons, make acetyl, throw it into the Krebs cycle, off to the electron transport chain, yay, ATP is made, okay? This is the description of how to catabolize, how to oxidize carbohydrates. In your body, when we eat carbs like starch or even shorter polysaccharides that you might find in beans and stuff like that, we break the chain of sugars down to monosaccharides. And if they're not all glucoses, we convert them to glucose. And then we throw them into this process. Now, we don't only have the ability to get energy from carbs, so that's a primary fuel. what are some of the other things that we can consume? What other organic chemicals in our diet get oxidized for energy? Carbs is one molecule in our diet that we use for energy. It's a primary source. What else do we have in our diet that we eat and that is catabolized for ATP production? Right, so carbs or sugar is one, right? We see that here. That's what we usually focus on. But also, yes, also lipids, also known as fats, also proteins. Okay, so I'm going to show you how these contribute to this process of energy generation very quickly, starting with this molecule. Now, what this is a 3-carbon backbone with tails that are long hydrocarbon chains that are hydrophobic. So what is this molecule called? It's got 3 of these fatty acid tails attached to a glycerol. What is this? That's right. This is a triglyceride. This is the storage form of fat in your body and in most organisms. And so it can be found in the environment if organisms eat other things. And so if you have this, what you've got to do is first take it apart. So you clip this bond here, here, and here. So you take the legs off the stool. Okay, you've got your glycerol backbone and you freed the fatty acids. The next thing that happens is these fatty acid tails are always an even number of carbons, and they get chopped into 2 carbon segments. This 3-carbon glycerol gets fed into part of this pathway. Where is there a place where a 3-carbon molecule already exists that if you just convert this a little bit, you could shove it into that spot? Where in this process of respiration is there a 3-carbon molecule that if you just take this glycerol and modify it a little bit, you could throw it in there? Yeah. So it comes in at the level of pyruvate. Then these fatty acid tails that are usually either 16 or 18 carbons long, that other lengths exist, but that's common. They get chopped into these 2-carbon bits. That process is called beta oxidation. And then these 2-carbon bits get fed into the level of, they're basically acetate. They get fed into this step right before the Krebs cycle. So actually lipid oxidation is fairly straightforward. And you can see that just taking one fatty acid apart gives you a ton of stuff to throw in here. Like 1 glucose allows you to crank the Krebs cycle twice, okay? 1 triglyceride may allow you to crank it 27 times. It's a significant increase in energy production. Let's talk about proteins. They're made of subunits. What are these subunits called? One of them is diagrammed here. That's right. So proteins are these chains of amino acids. First thing you got to do, hydrolyze them. So now you separate each of the fatty acids out. All the fatty acids have a central carbon attached to a carboxyl group, attached to an amino group, attached to a hydrogen, attached to a mystery group. And the first thing you have to do is throw the amino group away. So you deaminate the amino acid. Away it goes. Bye-bye. And then the rest of this has either 2 or 3 or 4 or 5 or 6 carbons, depending on what this group is. So is there a place where you could put molecules that are 2 or 3 or 4 or 5 or 6 carbons? If we look through this, we see lots of options. And in fact, depending on what specific amino acid this is, it could either go in, if it's 3 carbons, maybe at the level of pyruvate. If it's 2, at the level of acetate. If it's 4, 5, or 6, there's options in the Krebs cycle. Okay, now, in terms of understanding the next section, we need to ask a question. So you catabolize polysaccharides, long chain carbohydrates, lipids, and protein by eating them, chewing them, swallowing them. They end up in the stomach, and in the process, and they go through the small intestine. In that whole digestive process, the things that you eat are being acted upon by enzymes in your gut, basically at different parts, like in the mouth, in the stomach, and in the small intestine. Bacteria don't have a small intestine, so they're going to encounter these macromolecules and they've got to use them somehow, right? But they don't have a gut. So what they do instead is they take those same digestive enzymes and they spit them into the environment that they live in, and it digests the world. And then those bits, amino acid bits, the single amino acids, single sugars, and fatty acids come floating on back to the bacteria. So in order to do that, they use something called exoenzymes, meaning outside. Okay, so why do bacteria need exoenzymes? Well, because they don't have a gut. And they also don't have internal digestive organelles the way eukaryotes do. And then let's talk about the specific enzymes they're sending out there. So if you're a bacterium and there's a world that's filled with starch, you run into a cracker or a potato or something, what are you going to use to hydrolyze that starch down to glucose? And as a hint, it's the same enzyme we use. Yeah, and if you're not sure, putting a question mark is totally fine. You can also scan the chat and crib your answers from other people who were quicker off the bat. Yeah, so about half of you have already taken nutrition and maybe remember this from A&P. We use an enzyme, it's in our saliva, it's also in our small intestine, re-added after things go through the stomach, it's re-added by the pancreas. So we have salivary and pancreatic amylase. Bacteria have amylase too if they're able to use starch. And remember, amylase is an enzyme, it's encoded by DNA. So if you don't have the right instructions, you can't do this. But organisms that can actually sort of spit amylase into their environment and digest the starch, and then the individual glucose molecules can come floating on back and be imported one by one into the bacterium. Now, if we think about how we break down triglycerides to fatty acids, okay, think about what that enzyme is called. Same enzyme is used by bacteria. What is that name of that enzyme? If you're not sure, again, a question mark is a fine thing to add, or you can scan the chat and get your answers from other people. So it's something that breaks down lipids. So it is called lipase. And again, you can secrete this into your environment. And again, do all bacteria have lipase? Do you think? You'd be correct. Some do, some don't. So is that useful to us? Sure, because if we know what bacteria do and don't, and we do that test, we can narrow down what an unknown bacteria might be. Okay, we also break proteins down into amino acids. What's the general name for the type of enzyme that can do this? There's lots of specific names. What's the general name? So it's something that hydrolyzes proteins. And so yes, a good guess would be protease, or if it was something you already knew, good job. But proteases are secreted into the environment also. Okay. We're going to revisit this idea of exoenzymes a little bit later today in order to apply it to the practical setting of metabolic tests that we might do for an unknown. Right now, what I want to do, though, is I want to move on from hydrolysis and energy production, catabolic processes that break stuff down for energy. I want to move on to the carbon fixation metabolism that some organisms do that produce carbohydrates so the rest of us can live. And I'm going to start with photosynthesis, which is one you are more likely to have studied before, though not everyone will have studied it. And I want us to focus first on what we're most likely to know something about, which is what happens in a eukaryote. Now, of course, there is an organelle in eukaryotic cells where photosynthesis takes place. And what is that organelle? Put that into the chat. That's right. It is the chloroplast. And if you did the prep that I asked you to do, you'll remember one component of what I asked you to do is to draw a picture of a chloroplast and annotate where the different reactions within photosynthesis are taking place. So if you didn't do that or you don't remember, it's fine to put a question mark in response to this, but do put something in. But if you can remember in the photosynthetic eukaryotic cell, where the light reactions take place, where are the photosystems and electron transport chains associated with those photosystems located? We briefly touched on this when we covered chapter 4. Let's see if you remember. That's right. So the internal membrane of the chloroplast is called the thylakoid, and that is where you'll find your photosystems and electron transport chains that handle electrons. So what happens is that those photosystems steal an electron from water, producing oxygen. That's how oxygen is produced. And then they capture sunlight photons and use it to energize the electron, which then goes through one of the electron transport chains nearby. And that subsequent events are very similar to how ATP is produced in a mitochondrion. As electrons travel through the electron transport chain, protons are pumped across a membrane to create a gradient. And that gradient is then used to generate energy using ATP synthase. Where in a photosynthetic eukaryotic cell, which has a chloroplast, where are these hydrogen ions accumulating to form that gradient of potential energy? The thylakoid has two sides that are fluid-filled compartments. On one side is the the stroma, and on the other side is the thylakoid lumen, or the so-called interior of the thylakoid. Where are we pumping our protons in order to form this gradient? Again, we touched on this in chapter 4. Yes, that's right. We're pumping them inside the thylakoid. Now, the thylakoid is actually a pouch, this little circle. And so we're pumping it into the interior of the thylakoid. And that's useful because if we were to pump it into the stroma, we would acidify the stroma, which is where a lot of reactions are happening. So the dark reactions, the so-called Calvin cycle, what's happening in the dark reactions is that the light reactions have produced a lot of ATP and also some energized electrons. And then we take those two components and some CO2 from the air and squish them all together in the Calvin cycle to create organic compound that's eventually going to become sugar. These happen in a fluid-filled compartment. They don't happen in a membrane. And the dark reaction happens where? Again, it is a photosynthetic eukaryotic cell that has a chloroplast. So what fluid-filled compartment is where this chemistry is happening? That's correct. Yeah, it's happening in the stroma, which is the most interior fluid. It's analogous to the matrix of the mitochondrion. Okay, so this summarizes those things, right? The so-called light reactions are happening in the thylakoid membrane of a eukaryotic cell that has a chloroplast. The Calvin cycle happens in the stroma, and the place where the hydrogen ions are put is the thylakoid interior. Let's move on to a prokaryotic cell. Can we do photosynthesis in a prokaryote? Great. It'd be a pretty boring lesson if that wasn't the case. The answer is yes, but they don't have chloroplasts. They still have to have a place to put their photosystems and electron transport chains so they can do the light reactions. Where are those machines located in a prokaryotic cell? There's no thylakoid, there's no chloroplast, so where are they located? Correct. They're located in the plasma membrane. Now, the plasma membrane separates two fluid-filled compartments, just like the thylakoid membrane does. It separates the cytoplasmic interior of the cell from the extracellular space. Where are we pumping our protons? Are we pumping them into the cytoplasm, or are we pumping them into the extracellular space to create a gradient? Great. Yeah, we're pumping them outside. We don't want to acidify our cytoplasm and destroy all of our enzymes. So we pump them outside, we let them come back through ATP synthase, we generate some ATP, and then we go through the dark reactions, the Calvin cycle. Where is that taking place? Where's the Calvin cycle taking place? That's right. Okay, so this is a comparison we want to remember, and I'm now going to draw you. My favorite thing is to draw a picture. Start with a eukaryotic cell. Got a chloroplast. Here's the outer membrane, inner membrane, and then the thylakoid membrane, which is even more inside. Okay, so in the thylakoid membrane, we've got our photosystems getting electrons from water, getting energized by the sun, electron transport chain, pump those protons into the interior. Electron goes back through another cycle of being re-energized, just so we have energized electrons. That goes to NADP. Then we let those electrons out, or those protons out, to make ATP in the stroma. The stroma is where the dark reactions are happening, where we take carbon dioxide, the energy we made in the light reactions, plus the energized electrons from NADP to put together into 3 carbon molecules, 2 of which will be smushed together and make glucose. All right, if we compare that to a bacteria, let's say, then we've got our photosystems and our electron transport chains are in the plasma membrane. We're getting our electrons, energizing it with the sun, feeding it through the electron transport chains, which is pumping protons outside the cell, creating a gradient in the extracellular space, re-energize that same electron so that we can hand it off to NADP and save it for our organic molecule production. We get ATP production here, right, with our proton gradient. We put together ATP, energized electrons, CO2 to get a 3 carbon molecule. These are the dark reactions happening in the cytoplasm. A couple of these put together makes glucose. Something that I want you to strive for, because at least the Krebs cycle, and my hope is that maybe to some degree the Calvin cycle, are things you've heard of before. And there's lots of metabolic processes that you've been exposed to at some level or other. And in some classes, it's important to know the nitty-gritty details, like in a biochemistry class, for example, or even sometimes when you're first taught cell biology, they ask you for some details. But one way to tell that you've really integrated the important pieces of these biochemical pathways is to be able to sort of step back from them and say, okay, what's the big picture here? And I want to use that strategy now to help us feel more like we have a mastery of these biochemical pathways, that big picture perspective. And I want to compare the Calvin cycle to the Krebs cycle, because we can compare things like where they take place in eukaryotes and prokaryotes. Well, in a eukaryotic cell, they take place in different places, right? The Calvin cycle in a eukaryote is happening in the chloroplast stroma, and the Krebs cycle in a eukaryote is happening in the mitochondrial matrix. But that should tell you right off the bat that the stroma and the matrix actually have some similarities. And in a prokaryotic cell, both these processes happen where both the Calvin cycle and the Krebs cycle happen in the that's right, they happen in the cytoplasm. And do you remember the endosymbiotic theory? Remember that idea that chloroplasts and mitochondria were originally derived from bacteria. The matrix and the stroma are evolutionarily originally cytoplasms of a bacterium, right? So just bear that in mind. It's not sort of a separate concept. It makes sense, actually. Okay, if we compare the stroma and the matrix, they're both fluids that are around pH of 8. And the cytoplasm, right, is kind of a similar neutral-ish compartment where chemistry can happen. All right, now let's look at these from a different perspective, sort of from the what has happened chemically that we care about. If we think about what the Calvin cycle is doing versus what the Krebs cycle is doing, a simplistic comparison that's nonetheless sort of sophisticated is in the Krebs cycle, we're taking the breakdown product of glucose and oxidizing it, right? Oxidizing it down to CO2. We're moving electrons until all that's left is CO2. In the Calvin cycle, we're taking CO2 and we are adding electrons to it until it turns into sugar. So is the Calvin cycle overall an oxidizing or reducing pathway? In terms of the carbon compounds that we care about. Yeah, it's reducing. And again, I'm trying to cut through the jargon here because every field and subfield and subfield of biology and biochemistry and regular chemistry has all these like very specific terms, and they're not useless, right? We need the jargon to precisely define what we're talking about. But sometimes there's many terms for the same thing. And really what's happening in the Calvin cycle is that we're reducing carbon dioxide to produce glucose. And what's happening in the Krebs cycle is that we're oxidizing glucose and we produce CO2 as a waste. Okay. So again, the Calvin cycle reduces carbon dioxide to an organic molecule. And this, again, this is a chemical term. We think of reduction as being making something smaller, but it's actually the opposite. We're adding electrons to it and protons come along with that. So that's basically adding hydrogens to create a carbohydrate, C with a carbon oxygen H2 ratio. And in the Krebs cycle, we're breaking down an organic molecule that consists of carbon, hydrogens, and oxygens, removing the electrons, also the protons go away, and that leaves CO2. So if we think about sort of like the big picture purpose, Calvin cycle is important for something called carbon fixation, where we take CO2 out of the air, so an inorganic version of carbon that has no utility as a fuel, and we stuff it full of electrons, right? So that it turns into something, an organic compound that has lots of energy that other cells can eat. And in the Krebs cycle, we then turn around and take that glucose that's been produced by carbon fixation and break it all down again to get energy. So let's have a look at carbon fixation. There's the most well-known and well-understood one, photosynthesis. Most people have heard of this to some degree or other. In the microbial world, however, there's another process that's also a carbon fixation process that happens in a series of organisms that don't have the ability to do photosynthesis, in part because they don't live in a light-filled area. What is that alternative to photosynthesis in the dark corners of our world? That's right. Indeed, it's chemolithoautotrophy. So here is a little diagram of how chemolithoautotrophy works, and hopefully you can see the parallels with photosynthesis. An electron comes from usually a metal ion being oxidized. That electron goes through an electron transport chain, and it pumps protons, and those protons come back through ATP synthase, and ATP is produced. And the electron is then captured by an electron carrier. So then we have some ATP, we have some energized electrons, and we have some CO2 in the area, and we jam it all together in a series of biochemical reactions to create this 3-carbon organic compound that's then 2 of these are smushed together to make glucose. And that's basically what happens in chemolithoautotrophy. So it's quite similar to photosynthesis, except in terms of like, where did the electrons come from? And also, where did the energy, of course, come from? So why is this important? Why do we need chemolithoautotrophy? I mean, I've already said this a couple of times, so what is the answer? Why do, or maybe what I should ask is, what purpose do microbes that can do chemolithoautotrophy serve? Why is it that photosynthetic organisms can't just do the job and we don't need this extra pathway? That's right. There's life in places that have no access to sunlight, which means photosynthesis is just straight up not going to work. Okay, and what we're talking about is undersea, right, at the bottom of the ocean, for example, or underground, right, where light can't penetrate, or in a cave where there's no access to sunlight. In those types of environments, if chemolithoautotrophy wasn't something that microbes could do, there wouldn't be any life there. And that's a possibility, right? Maybe if this had never been invented, that those places would not have any life. But they do exist, and this is what's keeping them alive, basically. All right. So we're going to take our break now, and when we come back, we're going to try to apply this to chemistry, and we're going to be probably a little ahead of schedule. So I'm going to make a decision about whether we end class early or do a Q&A or what we're going to do, or if I forge ahead into genetics. But my inclination actually is to probably will end class a little early and do a bit of Q&A, especially for those of you who are trying to get some good head of steam on your unknown ID project. So we'll talk about that. But for now, we'll take our 10 minutes and we'll come back at 11:35 and talk more about this. I'll see you then. Welcome back, everyone. Let's talk about testing. So you'll remember these tubes, right, from our section on growth and from from lab last week, lab 3A. These tubes help us, they're called thioglycolate tubes, and they help us determine what about an organism. That's right. They help us determine their oxygen preference or oxygen requirement. Can they grow in oxygen or not grow in oxygen? Do they have the ability to grow without oxygen or not? Okay, so we can see all of the different possibilities. When we look at these tubes, they can tell us a variety of things. They can also, they're limited, right, in terms of what they can tell us. And we want to be really clear on what we can learn. Apart from being able to classify organisms, which is how we learned what we learned about and how we learn to use these in the growth section, which is like, what category is it? Is it, you know, a facultative anaerobe, aerotolerant, obligate anaerobe, what is it? We also can infer things, but we want to make sure we don't take our inferences too far. So when we look at these tubes, which organisms can we be really sure are capable of doing aerobic respiration? And these are the letters at the top, so you can just use these letters. Here's 4 letters. If none of them, just say none. If you think it's all, say all. But if it's some of them, put the specific letters you think. What does this tell us? Who can do aerobic respiration? Excellent. Okay, so yes, I think everyone immediately was able to tell that C is doing aerobic respiration because he's not obviously alive unless he's in oxygen, so he must be using it. But also, we can see that this one, right, this is also doing aerobic respiration, at least when oxygen is around. How do we know that's the case? Okay, how do we know that B is doing aerobic respiration and not just a bunch of other things? Raise your hand. How did you figure that out? How did you know it is also B, not just C? Jolene, go ahead.
A lot of growth is at the top of the tube.
Okay. And general question to the whole class. So you heard Jolene say, well, we can see there's a lot of growth at the top as compared to the bottom. So it does seem to be that something different is happening, right, in oxygen versus not in oxygen. Why is it the case that if you're doing aerobic respiration when oxygen's around and then something else when it's not, why would you have more growth in oxygen? What's special about aerobic respiration? What that explains this difference in growth? Put it into the chat. Yes. Remember, I earlier today just made this whole big deal about aerobic respiration gives you more ATP per glucose. So that means you're making more energy per unit time. And if you're making more energy, you can grow faster. And by growth, I mean you can do cell division faster, which means you're going to produce more organisms and you're going to visibly see more growth. Which of the organisms are capable of doing anaerobic metabolism? Great. Yeah, so again, everyone can clearly see that D is obviously doing something anaerobic, right? It's not even growing unless there's in the presence of oxygen. It's also true, as many of you noted, that A and B are also able to do some kind of anaerobic metabolism because when there's no air at the bottom of the tube, they're still alive. Now, are we able to be sure that we know what that form of anaerobic metabolism is? Could we say definitely, oh, it's fermentation? That is correct. All we can be sure of is we know who's not doing fermentation. That's this guy. Okay, everybody else is a question mark. And the reason for that is that fermentation is not the only game in town. Is it possible to be non-fermentative but still alive down here? If so, what could you be doing instead? Exactly. Since fermentation and anaerobic respiration are both possible, and since there's no way to visually distinguish them using this test, we don't actually know if they're doing fermentation or anaerobic respiration, or maybe both. Okay. Now, can we tell from these results who has the enzyme superoxide dismutase? Well, the answer should be yes, and you should be able to give me your answer. If you're not sure who it is, put a question mark or just give it your best shot. If you do know the answer, put your answer in, but even if you just want to guess, that's also fine. But which of the organisms in these tubes has the enzyme superoxide dismutase? Great. So remember that I told you that if you live in oxygen, whether you use oxygen in aerobic respiration or you don't, but if you're able to be alive when oxygen's around, then you definitely have superoxide dismutase. Okay, so those of you who said ABC are correct. And what we know for sure is this guy doesn't, because he's dead. Now, the other thing to remember is that there were other defenses against oxygen that some organisms have, things like catalase. And catalase or peroxidase are found in organisms that have to use oxygen for their metabolism because they create a lot more negatively charged oxygen to deal with. So catalase or peroxidase are possible. When we try to figure out who's got what, all we can say with certainty is who does not have catalase. Aerotolerant organisms don't have it. They don't need it because they're not actively putting electrons onto oxygen. They're just rarely, but still sometimes exposed to oxygen anions. So they just need enough superoxide dismutase to get that under control. It does produce a little bit of hydrogen peroxide, but not enough to be toxic. So they don't need any additional help. This guy clearly doesn't have catalase because he's dead. So it's only these guys that might, but they might have peroxidase instead. And again, we can't tell the difference. Okay, so that's why these are question marks. What about cytochrome C oxidase? What do you think? Yeah, so some of you have answers, some of you have question marks. Let's start with an easier question. Okay. The easier question is, who doesn't have it? Who do we know for sure does not have cytochrome C oxidase? Great. So most of you picked D as being the guy that we can be sure of. In fact, we can be sure of two of these. We also know that A doesn't make it, and that's because an aerotolerant organism doesn't use oxygen. Even though it's living, it's doing whatever it's doing down here is the same thing it's doing here. It's not using oxygen, so it doesn't need the enzyme to put electrons onto oxygen. These other two are more complicated. So those of you who are feeling uncertain, well, maybe you are feeling uncertain for the right reason, which is actually, if you have cytochrome C oxidase, then you can be sure that an organism can do aerobic respiration. But if they don't have cytochrome C oxidase, it's actually not for sure that they can't. And that's because cytochrome C oxidase is just one of several enzymes that can do the same job, just like catalase and peroxidase can do the same job. So not having cytochrome C oxidase just means that either they don't do aerobic respiration at all, or they do it using an enzyme that's does the same chemistry but has a different name. So hopefully that's not too confusing. It is true that most obligate aerobes have cytochrome C oxidase, but some of them have something else. And it is also true that a lot of facultative anaerobes are actually oxidase negative, although some of them are oxidase positive, and that some of them have this one and some have a different one. Let me just pause there for a second, let you absorb that. Did that make sense? Yes, no, or kind of? Yeah. So I think that, some of these ones with question marks can be a little tricky. And it is starting to get into the nuances of stuff, right? The sort of things that you just know as a content expert that are subtle, and I won't like nail you to the wall with these on exams. I will lean into the more obvious ones, but I'm bringing this up because are any of you, for your unknown ID, facultative anaerobe? Did any of you, in the process of getting to step 2, okay, if you haven't gotten to step 2 yet, as a facultative anaerobe, did any of you have to do an oxidase test next? Okay. If you're really paying attention, you might feel confused by that need for oxidase. You might also feel confused by why couldn't we do an oxidase test to find the answer to oxygen preference. Okay, and this is the reason. The oxidase test specifically tests for this enzyme only, and not for all enzymes that can do the chemistry that this one can do. So an organism that doesn't have specifically cytochrome C oxidase might still have the ability to do aerobic respiration with a different enzyme. And that's why there are facultative anaerobes that are oxidase negative and some that are oxidase positive. And if you are a gram-negative rod and you did the thioglycolate test and got this result, and now you're facultative anaerobe, you still have to do an oxidase test to find out which category you are in. Does that help at all in terms of understanding why I might be kind of introducing this slight subtlety? I just don't want anyone to wake up at 2 o'clock in the morning and go, wait, what? That doesn't make any sense. Instead, I want you to be here today going, wait, what? You don't make any sense. Okay, so if this is something that continues to trouble you, please let me know. All right, let's move on though. I have more things to talk about. Now, are we able to state definitively who has this enzyme nitrate reductase? Remember, nitrate reductase is used in one possible form of anaerobic respiration. All right, so many of you are saying no, which is the correct answer. Raise your hand if you want to explain why you're so confident that it is no. Why can't we be certain who's got this? We can be sure who doesn't. We'll get to that in a second. Sam, go ahead. You popped your hand up and you answered first. Tell us why we can't be sure who's got nitrate reductase. We know some of them might, but how come we can't be sure?
Because there's multiple different things that could take the place of that.
Right. Instead of nitrate reduction, maybe we do sulfate reduction or carbonate reduction. Yeah, exactly. We don't know for sure. And in fact, maybe we don't do any form of respiration and the guys that are living down here are doing fermentation. This test doesn't tell us that unless we do a different kind of test, which we can do. We could do a nitrate test. We could get this and go, I'm curious, and take our facultative anaerobe or our aerotolerant guy or our obligate anaerobe and say, hey, do you do nitrate reduction? Here's a nitrate test. Let's find out. Okay, and that's relevant because we're doing nitrate tests this week. All right. The only thing we can be sure of is this guy does not have the ability to do nitrate reduction. That would be a form of anaerobic respiration. So we know that he's going to be nitrate negative. And again, I just want to be very clear and honest about this. I am oversimplifying the situation. I am making the diversity of microbial metabolic pathways smaller so that we can hold them in our hands in this course and not lose our minds. Okay. So this is going to connect us back to the growth section, which is why I do it first. But we see 3 broths here. These are just pictures of broths, hypothetical pictures, but we notice it's that there's a lot of growth here, a medium amount of growth here, and there's no growth here, okay? So I'm giving you the option of, did something grow really well, kinda, or not at all? So this is kind of a multiple choice question. 2 different mesophiles, okay? So 2 species, both mesophiles, grown in oxygen. One is a facultative anaerobe, the other is aerotolerant. We can assume they're not fastidious and that the broth that we are growing them is sufficient for life. Which tube of these three possible options represents the facultative anaerobe if both are here somewhere? Okay, there's three tubes. Two of them represent real things and one is just a distractor. Which of these tubes most likely represents the facultative anaerobe? Great. Why is that? Raise your hand if you can explain how you know the answer. Most of you picked B. How do we know that that's the answer, which is true? Jolene, go ahead.
There's more growth in B compared to A or C, which indicates that it grows better in oxygen.
And what metabolism is it doing? Anyone else want to take up the slack here? Jolene's put in the heroic effort of answering the initial question. So my follow-up question is, she's right, there's more growth here. What type of metabolism is happening in this tube? Sam, go ahead.
Mostly aerobic respiration.
Aerobic respiration, right? And that's why we see this larger amount of growth. Okay, great. Let's look at this same tubes, different scenario. Okay, let's take one species, a facultative anaerobe. We're going to inoculate it to two tubes. We're going to grow both tubes for 8 hours at its favorite temperature, but one we're going to provide air and one we're going to deprive it of oxygen. Which tube best represents the tube that's grown with no air? Put your letter into the chat. And which tube represents that same species grown in air? Right. So the tube that best represents the tube grown with no air is A. And the tube that represents the species grown in air is B. And again, why? Because in air it's growing better, and in the absence of air it's still growing, but not as well. And that is because here it's able to do aerobic respiration. Here it's doing something else. Fermentation maybe, anaerobic respiration maybe, both, we don't really know. Okay, we know it's not this because they're both able, it's a facultative anaerobe by definition, it can grow without air. Okay, so this section on metabolism is timely and important for you at this stage in the game for the unknown ID process, because you're going to submit your unknown to a variety of tests to try to figure out who it is, and really at this point, mostly to figure out who it's not. And I want to kind of go over the universe of tests that you can run and connect them to the class material. So you can test your organism for its ability to grow at various temperatures just by asking for a broth at temperature X. Can I get a broth at temperature 11 degrees Celsius? You can ask for growth at pH of whatever. You can ask for growth in a particular percentage of salt. You can also ask for a thioglycolate test to determine what its oxygen preference category is. You can ask for a catalase or oxidase test to find out if it has this enzyme or that enzyme. You can ask for a nitrate reductase or nitrate broth tube to find out if it's able to reduce nitrate. You can ask for a different type of test called Kliger's iron hour to find out if it is able to reduce sulfate. And you can ask for a variety of plates, like a starch plate, a milk plate, or a lipase plate to find out if it's got the ability to hydrolyze carbs, proteins, or fats. Basically, does it secrete an exoenzyme? You can find out if it's got the ability to ferment a variety of carbohydrates using a phenol red fermentation test. And there is another kind of carbohydrate utilization test that we are not running this quarter. So I just want to acknowledge the existence of this since it's in the notes and places, but you will not ask for this one. Now, this week we're prepped and set up to do these types of metabolic tests. And I want to show you, sort of preview what they're going to look like and help us to connect this to the material. But before I do that, I just want to give you all a warning or heads up about this week. It's nothing about you or anything dangerous. I may have gotten sick. I'm not sure. I am getting tested today. My daughter has been sick for a week and I've been fine, and she's tested COVID negative, but I'm starting to not feel super awesome. So there is a chance, and I don't know exactly when we're going to decide this, that I may have to cancel lab tomorrow. I hope not. And if I can avoid it, I will avoid it. But in that event, I will send you instructions. What will happen is we'll make a slight adjustment to the lab schedule. And you'll do the digital part of this week's lab on time so that you have that information and it's like still supporting your process in terms of conceptual understanding of the course and your unknown ID process. But we might end up having to delay the physical performance of some of these tests until next week. So I will keep you posted and let you know exactly as soon as I can. But I just wanted to let you know that possibility, which is not really useful information since I don't know the answer yet. So with that said, let's move forward. So one of the things we'll do in lab is a catalase test. It's quite a simple test. Basically, you take a glass slide, you rub some cells onto it, and you add the reagent. And from your prep for lab and for this week, does anyone know what reagent you'll be adding for a catalase test? Any thoughts? Remembering what catalase does. That's right. Good job. Yeah. So catalase is an enzyme that takes hydrogen peroxide and makes it harmless by turning it into water and oxygen. And so you add your hydrogen peroxide to the little dry smear of cells and you see what happens. Okay. And obviously, there's the positive result, and this is a negative result. So let's suppose you have a facultative anaerobe and an obligate aerobe. Like one of these samples is the one and one is these samples is the other. I earlier told you that if you knew these categories, you couldn't be sure about catalase. But if you know that you have one of these and one of those, and this is what you get, who's the positive? If you have an obligate anaerobe and a facultative anaerobe, who's testing positive for catalase? That's right. It's obviously A, because the obligate anaerobe has no defenses against oxygen, so it doesn't have catalase. And I'm bringing this up for two reasons. Again, I want to support this. Many people, when they're doing the dichotomous key for the unknown ID, they try and they get to that decision point that says oxygen preference. They try to do a catalase or an oxidase test for it. And that is not the best test because there's so much ambiguity in terms of how to interpret the results. However, it is a very useful test to distinguish subgroups of organisms. Some are catalase positive and some are catalase negative. And you can conclude some things from it if you have some information already, okay? So if you have two organisms and you know for sure that one's a facultative anaerobe and one's an obligate anaerobe because you already did the thioglycolate test, then you're going to know what their catalase results are going to be if you do this and you see that one's positive, if that makes sense. You're not going to know it until you do this, but if you do this and you've got two and you know one of them has to be positive because that's what you got, you know who it is. So hopefully that was not too confusing. Here's the oxidase test. You're going to do this in lab also. It's pretty fun. So it's basically a piece of paper soaked with a reagent that cytochrome C oxidase reacts with. The reagent's colorless, but as cytochrome C oxidase is there, it turns the colorless reagent into a purple product. So when you get this result, okay, let's say you just did it for your unknown out of the blue, like you knew nothing about the unknown, you just did this result. Then what you can conclude from this test is what specifically and narrowly can you be 100% sure of? That's right, Sam. That's right, Libri. That's like Quinn, Jolene. Yes. What you can be sure of, if you know nothing else, is that it's cytochrome C oxidase positive. Okay. Now, as it happens, once you know this, you also do know that it can do aerobic respiration, but it's not possible to go back the other way. Like, let's suppose you had gotten a negative. Would you be able to be sure that it's not doing aerobic respiration? No, because there could be a different enzyme that's not cytochrome C oxidase that wouldn't turn positive on this test, but still could add electrons to oxygen. And that's how diverse microbes are. So does that make sense? Yes, no, kinda. Okay. We're starting to feel a little more comfortable with the ambiguities that I have introduced into the things. Okay, good. So these tests are helpful in that they can distinguish groups of organisms from each other. They're helpful in terms of they can give us very specific answers, and sometimes we can infer more things from them. But it is a mistake to have logic always going both ways. You have to be careful. So when this is positive, we can be sure that the organism's able to do aerobic respiration. This alone wouldn't tell us exactly what its oxygen preference is. This wouldn't tell us the difference between an obligate aerobe and a facultative anaerobe, both of which can grow in oxygen and use oxygen. And this result doesn't tell us that for sure something can't do aerobic respiration. So we want to be careful in terms of what we conclude from these by really understanding what they're telling us. Now, another test we're going to do in the lab is the nitrate test. What does this test actually tell us? Can this organism do what? Or does this organism have enzyme? That's right, Quinn. That's right, Jolene. Indeed, yes. Y'all did your lab prep, wonderful. Excellent. Okay. So the nitrate test says, can you reduce nitrate? Meaning what? Meaning what? What does it mean to reduce nitrate? Like what's happening chemically to the nitrate? Okay, good. Lots of different answers in the chat that are all correct. Okay. What it tells you is that the nitrate is having electrons added to it. Okay. And the first thing that happens then is it turns into nitrite. Sometimes it also then is converted into other things, though not always. It depends on the organism. But the first thing that happens is it's turned into nitrite. Hold on one second, guys. I need to fix something here. All right. So what you might recall from your prep, and I'm not expecting that you will have a solid grasp of this. Okay, so if you're not sure, you get this wrong, then it's fine. I'm not going to hold that against you. But what you might remember from your prep is that the nitrate test is a bit complicated in terms of its interpretation, and that is because while there's only one way to be negative. There's two ways to be positive for this test. That's because, as some of you noted, some organisms turn nitrate into nitrite, and some of them turn it into something else. Nitrate and nitrite can both be tested for with different chemicals. And then the absence of both those things tells you something. So it isn't actually as straightforward as some tests where one color means one thing for sure and one color means something else for sure. You have to know where in the test the color happened. So what you'll notice here is that there's 3 red tubes and 1 clear tube. First of all, without any further information, so sort of ignore these little annotations right now, without any further information, is this clear tube positive or negative for nitrate reduction? Did this organism reduce nitrate or didn't reduce nitrate? Great. So yes, most of you have correctly stated that Pseudomonas aeruginosa is nitrate positive. So being clear at the end of the experiment does mean that actually what it really means is nitrate and nitrite are both not found. And the only reason that that would be the case is if nitrate, which was provided in the tube at the beginning, was converted to something else that's not nitrite. So then we know, okay, it is reducing nitrate, but not to nitrite. Well, not eventually to nitrate. We'll talk about this in lab some more. Now we're left with these 3 red tubes. One of these red tubes is negative for nitrate reduction, and the other 2 are positive for nitrate reduction. Any guesses as to who is negative for nitrate reduction? Jolene, did you have a question?
Oh no, I was just gonna answer
Oh,
instead
you were
of
gonna
typing in the chat.
Okay, go ahead and answer.
Okay. So Streptococcus pneumoniae.
Great. And everyone else in the class agrees with you. And so it's maybe you were tipped off by the fact that it looks like a little bit different in color. But you know, if it was just a one-off test and you just did the test and you didn't have anything else to compare it to, then the red would be like a bit harder to interpret, unless you know this information, which is that it's a 2-step test, and turning red in step 1 means definitely positive. That's what happened here. And turning red in step 2 means definitely negative. That's what happened here. What happened here is that nitrite was detected. So if you're still feeling a little bit like, what? After that, we will clarify this in lab, but I just wanted to give you that heads up. Okay. So what we know for sure is that these 3 tubes on the right are positive for nitrate reduction. So this is a warning that the color alone doesn't tell you anything. You actually have to know a little bit more information. And the tube where nitrate and nitrite have both disappeared is this one. Okay. And this guy over here was not able to reduce nitrate at all. All right, now we get to the exoenzymes. This is a milk plate, and it's, as you might have guessed, it's got milk in it. Unfortunately, there's also a recipe for chocolate agar, and there's no chocolate in it. So, but this is a milk plate, and you can grow organisms on it. And this is a differential plate. So you can see that there's like something grew here and something grew here. One of these organisms produced the exoenzyme caseinase, and that digests the protein in milk that causes it to be milky. And exoenzymes, remember, they diffuse away from the streak. They diffuse away from the organism. They digest the environment. Which organism is positive for caseinase? Yeah, I think it's pretty straightforward. The one that has a clearing around the streak, the milk's been digested. We can see that. And again, why are exoenzymes important for bacteria? Well, they don't have stomachs like you. So this plate is a picture that I stole from the internet and realized after the fact that it had been mislabeled by the original person who posted it, and I haven't fixed it yet. So sorry. I should make this into one of those like is this true or false, right? I should make this into a true or false. But in any case, what you can see is a plate, the lipase plate has a dye, and the dye is an oily dye. It's a lipid-based dye, okay? And this particular dye is blue. You could have a different color. There's a red plate too. You grow your organisms on it, and then you see whether they secreted an enzyme that could digest that dye. And of course, who is lipase positive here? Is this plate true or false? Let's do it anyway, just for fun. Is this true or false? Is this picture an accurate statement, or is it a misleading? This bacterial plate is gaslighting us. That's right. It is the bottom one. And what class of macromolecules is being digested on this plate? That's right. Lipids, right? Lipase plate. So some organisms have the ability to digest lipids, some don't. Okay, now remember we can also hydrolyze or digest starch down to its glucose component. This plate, two things grew. Here's the growth for one, here's the growth for the other. Now, for the starch plate, it's got starch in it. Starch sometimes is a bit milky, sometimes it's more clear. So we don't look for clearing on the plate itself because that's not very reliable or always easy to see. Instead, what we do is we add a reagent called iodine to the plate, and iodine has the ability to bind starch and turn brown. So everywhere that starch is brown. Everywhere that starch has been hydrolyzed will be clear. Which of these organisms hydrolyzed starch? Okay, that's great. The one on the left, clearly, because there's a clearing around the street where their starch is gone. And what was that enzyme that they were using to do that again? That's correct. Good old friend amylase. I don't know how you feel about sharing enzymes with bacteria, but there's a lot of examples of that, of course. Once we realize that there's so many shared cellular processes, it makes sense. I think the thing that's the craziest to me is that we share an enzyme with a virus, an antibacterial virus, one that doesn't affect us at all. So we make an enzyme that's that some viruses also make. And that's pretty crazy. All right, so that's the end of the metabolism stuff that I'm going to talk about today. So I'm just going to end the recording and switch to the next topic for a second here.